wanted to thank Dr. Shantz and the whole team here for inviting us. And I was saying to Dr. Shantz at the break, this is by far the most satisfying Children's Environmental Health Center event I've been to. And we have a lot of very fun, very large meetings in exotic places like Rome and Sao Paulo. And then we have very small meetings with our project officers. But there's something about the interchange that's been really fascinating. And I was going over my slides break as well, and I was saying to Dr. Dolinoy, I said, I don't think I know what we're doing anymore, because she looked at me startled, and but what I meant was it's like the context is such, it's brought a lot of new questions for me about what could be the underlying mechanism. So in that spirit, I did bring some data that's kind of hot off the press that we for sure don't understand, but maybe you'll have some insights. So I'll just take a few minutes and describe our center, and I think you'll see some common themes. Our overall research goal is to look at how mixtures to endocrine disrupting chemicals might alter the trajectories of physical growth. So we're particularly interested in adiposity, but we also in the background are looking at height and changes in lean mass as well. Trajectories of sexual maturation, and in our Children's Center, for which we're re-recruiting subjects right now, we're actually able to get longitudinal measures of sexual maturation, which is very important because the tempo varies, has a wide variability anyway. And then we, of course, think we may see both retardations in maturation as well as accelerations for other toxic kinds. So it's kind of a mixed up mess and a challenging one that we're enjoying looking at. And the last sort of set of outcomes that we're looking at in the human models is metabolic homeostasis. So I'll talk more about that when I describe one of our projects. What else to tell you? So in the morning, I know you're looking a lot at BPA and phthalates, and we are as well, and that's been a common interest across our two centers. And then you'll also see the two sensitive periods approach. So we're also very focused on what is the role of in utero exposures and also exposures during the transition from pre-adolescence to adolescence, or the period of the pubertal transition, which is, as Dr. Shantz said, it's very dynamic. And we haven't figured out how to do this yet statistically, but we're particularly interested in whether that sort of second hit or second metabolically active period actually brings into, you can see visibly some of the effects that might have been set up in utero. So that's one of the things we're trying to wrestle with right now. I was talking about wrestling. Yes, we were talking about wrestling. Wrestling, I think, is kind of the operative word for a lot of these. And in Spanish, we collaborate with the National Institute of Public Health in Mexico. The word is luchar. So does anybody speak Spanish here? Yeah, so we're just, struggling and grappling with these data issues. So, thanks to Lisa Marshallowitz, who is responsible for these graphics, and they have stood the test of time. I wanted to briefly mention a couple things about the organization, because I think there's so many interesting aspects of the centers. We have, for our center, I was PI of the Schwanning Center, the Cognitive Center, but we went to a large center, Basanta Patnamadaban, who is a pediatric endocrinologist who works more with animal models and to some extent human models, as you'll see. She joined, so we're co-PIs together at the center. And one of the aspects I did want to emphasize that I think you see also in the Illinois Center is the importance of career development. So we had 3 people who are graduate students and very senior postdocs drive all the way over just for the the interactions, the chance to present their work. And across the children's centers nationwide, this is a huge emphasis. And I find it extremely rewarding. And just like the community centers the community core, engagement core is a required element of our center, we also have to have a very strong commitment to training and development of junior faculty and development of students. So I see that as particularly thriving here in Illinois, which is great. The other thing I wanted to mention for our center that's been an area we had to really aggressively develop even more than usual is our data management and modeling core. And I think you'll see from some of our data that we have moved heavily into just a toy with our mental health. We have moved into high dimensional data. So not only epigenetics, but also metabolomics, which is a newish technology that we can use to look at metabolic outcomes. And the person who heads that is Peterson, and he is an expert in sort of high dimensional statistical modeling. We're talking, for example, over 9,000 measures of a single outcome, those kind of challenges. And he actually enjoys that, which is great. Part of the reason I want to bring it up too is he did just get an R01 in this area, and so he's doing a lot of work in developing methods that he would like to be able to disseminate more widely to the other children's centers and even develop statistical packages. So if there's something you see in our talk that could be a potential benefit, let us know. So research project, we have 3 research projects, and 2 of the projects are in humans. Research project 1 is led by John Meeker, and I'm also involved in Joyce Lee, who's a pediatric endocrinologist. I think John was here last week. I don't know if you talked about this, but I'm guessing you talked about some things related. This is in the Mexico cohort, which I'll tell you about in a minute, and is basically a follow-on of our formative centers. So we're looking not only at BPA phthalates. I didn't mention early on, we also are sort of equal opportunity employer when it comes to toxicants. So it turns out metals also are kind of bad boys or bad girls in terms of endocrine, sort of perturbing certain endocrine processes and or related metabolic pathways. So we also are looking at cadmium in the expanded sample. And I think that's all I'll say there. The one thing I would also mention, I noticed Susan, Dr. Cork, there she is. So we, after a lot of agonizing, decided we should try to get a physician-measured Tanner staging of sexual maturation. So we actually were able to do that. So we've been looking very closely at self-reported sexual maturation, physician-observed, and then how that relates to hormones that correlate with secondary sexual characteristics. And the good news is actually that self-reported sexual maturation with some help from the mother is actually quite valid. So we're trying to get that paper out. Research Project 2, which is the one I'll share some results, I co-lead with Vasantha. And here we're looking at these lead, cadmium, BPA, and phthalates during pregnancy and during puberty. And we're interested in how they may affect certain epigenetic mechanisms. And I think Dana's going to talk a tiny bit. I don't know if she's showing anything, Jackie's epigenetic data, but I'll leave that to them to mention or to follow up with you in the question period. And then we also want to look at how that may affect metabolomics biomarkers and components of metabolic syndrome risk in adolescents. So again, we're interested in what happens during pregnancy or in utero, what happens concurrently as you're going through the peripubertal transition, and then how does that affect your metabolic risk in peripuberty, just because those are the data we have. I won't be able to mention the new cohort. This is Vasantha's cohort that she and Dana started, and this is a Michigan-based cohort that's growing. It's recruited in pregnancy, is that correct? Prior to pregnancy. And then women are followed through pregnancy, collect their placenta, and characteristics of the newborn. Right now, there's no follow-up of the children, but it's a hope. So this is a really interesting, very intense pregnancy cohort, and then the element cohorts that I'll tell you about. The other thing we're very interested in, and I'll only say a tiny bit, and then I think Dana will talk more with you, and Lisa talk about diet in her poster. We're very interested in how diet, both during pregnancy and in adolescence, could modify the associations. And if you start to think about metabolic risk and related outcomes, you can realize that diet could push it either direction. You could have a good setup from a toxicant point of view. You might be countering it with certain methyl donors, and then bam, you get a lot of sugar-sweetened beverages in adolescence. It washes it all out. That's why we're glad we have a great statistician. So we're just entering into diet, and we're recruiting a couple of new people to work on that because it's sufficiently complex. I'd be happy to answer questions on that. I think research project 3 is led by Dr. Dolinoy, and this is in the mouse model, and I think I'll let her describe that too. But they base very theme in common with the UIUC Center is that we are very interested in learning from animal models and human models and dovetailing those. And then we need Barbara's help in kind of translate it. The Goody sisters are very cute, very cute. So our research setting for Project 1 and Project 2 that comprises the Mexico cohort, many of you may have heard, or at least some of you will be very familiar with this. This was actually started in the early 1990s by Howard Hu, and we Mauricio Hernandez, a younger nutritionist, Karen Peterson, and a younger nutritionist in Mexico, Teresa Gonzalez de Costillo. We all met in Cuernavaca and cooked up this idea, which was basically Howard and Mauricio were very interested in the resorption of bone during pregnancy to make calcium available to the fetus and whether lead would be released as part of that process. So that led to a whole series of funding, and now Howard's gone on to be Dean of the School of Public Health at Toronto. He still has his hand in research in the cohort, but at Michigan we have all the biologic samples, and these mother-child pairs have been followed since the mid-90s into early 2000s. The oldest children are now 20, and I think we have 3 infants already of the kids in the study, and we're trying to figure out what to do with them. This is the field staff at the National Institute of Public Health in Mexico with our project coordinator, who some of you will meet when you come to the October meetings, Lindsay. Very familiar. And then I wanted to particularly single out Mara Tellez Rojo, who was Mauricio's doctoral student many years ago, and she's our Mexican PI, and some of you do know her. So we had 3 birth cohorts that were recruited starting in the mid-90s, as I mentioned. The latest was up to 2005. There's actually a 4th element cohort that Bob Wright maintains at Mount Sinai School of Medicine, and Andrea Baccarelli has an R01 looking at primarily epigenetics in relationship to stress and some of these exposures. And what else to tell you about it? Basically, we're in the process of re-recruiting now. I was really relieved to hear you have about half your sample recruited, because that's where we are. I'm getting, you were just into year 3. I'm like, oh, please, 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 can we just recruit the sample and get enough analyses done we can get refunded 3 years from now, you know, the life of academics. So we can have a support group. It's like, yeah, I think I just got an email that we're up to 225 or something. I don't know where it goes. And the re-recruitment is done in Mexico City by the staff that you just saw. So Deborah Watkins is a postdoctoral fellow that was at Brown and now is in John Meeker's lab, and she writes papers and does a lot of research very rapidly, and she's also very interested in metabolic outcomes. So she is the one who sent me with my little lunchbox full of some new data on phthalates and BPA, and particularly we're starting to look at how puberty might modify some of the associations with metabolic outcomes. So I'll share those. Why do we care about EDCs and metabolic syndrome? It's like it's preaching to the choir, I think, here. Metabolic syndrome does affect about 30% of obese adolescents, but what's interesting actually is metabolic syndrome risk is not particularly well defined, and there's a lack of consensus about what it means in children and when it shows up and how early it shows up. So what you'll see here is because we don't have the numbers to have a high prevalence of meeting all the risk factors for metabolic syndrome, which you see listed here. So it's a high waist circumference. We're very interested in the central fat distribution in combination with at least 2 of the other things that are on this list here. So elevated systolic blood pressure, elevated triglycerides, lower HDL cholesterol, and or high fasting glucose. And what we're finding in kids is not everybody will meet all those criteria to have full-blown metabolic syndrome risk. So we're developing some other analysis strategies to handle what if we see lipids going off track. And I think what you'll see in the results is things seem to be affected differently by different toxicants and exposures in different time periods, both in boys and in girls. So I'm, in the course of this day, becoming less convinced we actually want to look at metabolic syndrome as a constellation. So you know what you think. I won't go into the data here, but why would we want to look at this in the element cohort? The bottom line is that we started to look at what we were learning about toxicant exposures in the element cohort as the kids were aging, and we started to see that some evidence that the effects of the exposures were varying. So in utero exposure seemed to have a different effect, or even sometimes a different direction of effect than concurrent exposure in adolescents. We were seeing all sorts of evidence that the effects of exposures were different for boys and girls. A theme in general, I would say, for cardiometabolic risk is that girls seem to be impacted more than boys, with a couple exceptions. So I'd love to hear people's thoughts on that. And the other, the BPA and phthalates, this is a poster Tiffany Yang, who's now at University of Aberdeen, presented a couple years ago at ISEE. This basically also presents sort of a really complex picture for phthalates that I think won't surprise anybody in this room, but it really seems to be affecting fat distribution. So not only waist, but also tricep skin fold. And I personally teach a course on growth and maturation, and I'm ridiculously enthusiastic about that topic. And one of the things that's really interesting about tricep skin fold is people are always looking at central distribution in relation to metabolic risk, is it goes down dramatically in boys as they go through puberty, and their lean mass increments dramatically as well. And I think we haven't been looking enough at changes in lean mass in the picture of anti-androgenic hormones. So that's another topic I'd love your insights on. So here I'll actually show you data which are from our P20 center. We are getting some of our new outcomes, but it still is on our initial sample that we had in our formative center. So we're looking some evidence of, I'll just show you some snapshots, BPA, phthalates, and lead exposure, and what their effect is on fasting glucose, serum lipids, and also leptin. So leptin is a hormone that's that's produced by adipose tissue. So it's of great interest to us here, and it's a much sort of a more direct measure, which I say it's probably more closely linked to mechanisms, and we might learn more from it than we would from body mass index. So something to keep in mind is, it turns out even though we use body mass index to classify whether children or people are overweight or obese, in fact, BMI only correlates with adiposity in the top end of the distribution. So that's why we like to look at leptin as well. So for these data, I'll show you now about 250 children that we re-recruited. So here it is, roll forward. It's not 20 years later, but it's 8 to 14 years later from when they entered the study 3 years ago. We looked at exposures in 2 time periods, and then we have the advantage of having a lot of archive samples. I would say our study design, its different outcomes was very comparable to the New Bedford study. It's that type of a cohort study where you have the good fortune, and sometimes the vexing problems of a very old biorepository that you can go back and look at early measures of exposure, as long as the freezers stay on. See Susan and I at lunch if you want to talk about freezers. Let's see, so in terms of exposure periods, for these data, which are more pilot data, we were able to analyze third trimester urines for BPA and for phthalates. And those of you in the room who work in this area, you know you need many more measures than one measure in pregnancy. So we did, with our additional funding, those, we looked also at first and second trimester urines, but we don't have those results here. And then we also looked at BPA and phthalates in concurrent urines of the adolescents. We have a number of lead exposure measures in this cohort, and I'll only talk about the cumulative measure of exposure in pregnancy, which is actually bone lead of the mother that we measure at 1 month postpartum, and with a KXRF machine. And then we'll also talk about concurrent lead exposure. But we do have this advantage actually of also having trajectories of exposure for this one particular exposure. And then the outcomes I mentioned here. So the in passing. So we're looking then, instead of at metabolic syndrome risk as a constellation and meeting the entire definition, we're looking at children's cholesterol levels in their blood, their HDL cholesterol, or so-called good cholesterol, LDL, or so-called bad cholesterol, triglycerides, glucose, leptin, and then untargeted metabolomics, which is how we're using ourselves these days. These particular analyses are really straightforward, just linear regression to model whether the exposure in pregnancy or the exposure in adolescence affected metabolic risk, controlled for some covariates. And the outcomes here, some of you have seen some of these, a couple of these slides, we've done some reformatting, are presenting the percent change in the outcome for an interquartile increase in exposure. We'll only briefly mention a couple of things about samples. So I was interested that the New Bedford cohort was 15 years old on average in the data that you showed, Susan. And here, for these data, the children are on average 10, the standard deviation of close to 2 years. So they are just sort of in the early half of peripuberty, but some of them already, about a third already have experienced menarche, for example, among the girls. So we're kind of right in the heart. And if you look at the BMI Z-score, it's close to 1. So that means that, like the US, although these children are not quite as heavy as children are in the US, their mean body mass index Z-score is about 1 standard deviation above where it should be. The blood levels are higher than they are in the US overall, but they're about the same as they are in Detroit, Cincinnati, Boston, New Bedford, a number of the major urban areas. So I've had to write a lot of letters to NIH and EPA justifying why we would do a Mexican cohort. So that was one of the things that we did. And I didn't bring the data with me today, but the distributions of phthalates and BPA are somewhat similar to NHANES. There's a couple of the phthalates that are higher in Mexican women, possibly related to cosmetic use. So this one's, let's see, some of you will have seen this before. So this is the only slide on BPA and the serum metabolic markers at age of 14 years of age. And the bottom, so on the top what we have is, this particular one is our next, we have boys here. I only showed the outcomes for which there was something significant going on. So serum cholesterol, LDL cholesterol, triglycerides, so these being elevated blood lipids, fasting glucose, and then leptin, which is associated with adiposity. So we'll come back to boys. We saw no effects of EKI lipids in boys, but we saw an effect in girls, so a 7% increase in total cholesterol in girls related to mothers' pregnancy levels of EPA, and then a pretty large percent change for a child increase in EPA of the mother, almost a quarter, 25%, 22% in triglycerides in girls during adolescence. So we're actually right in the middle of trying to write this paper. We put it off for a couple months because it's kind of vexing, and the place at least I'm looking is there's so many fluctuations in mother's lipid metabolism in pregnancy, but what I don't know is why that would affect girls more than boys. So if you have any ideas, let us know. And then this is a very interesting effect here. One of the issues we're struggling with is this confounding of leptin and BMI, which don't relate perfectly. So when we take BMI out of this model, this association goes away. But when we control for BMI, which is really controlling for your overall size, including your lean mass, and boys, just remember, I told you we're getting very lean in this period. So once we control for their overall mass, so to speak, leptin, which is produced by adipose tissue, is associated with their concurrent BPA exposure. Could all just be diet. Maybe they're just eating more and it's sort of somehow in the canned goods. So these are really complex and annoying and fun issues that we're dealing with right now. Okay, I'm gonna laser it a bit because the time go. In terms of urinary phthalates and metabolic markers in boys, the bottom line here, and this is the one at the break, I was trying to find Dana, I said, I think I mentioned it, Jack, I have no longer have any idea why this may be true. So the bottom line here is that phthalate exposure, primarily concurrently in boys, and there's different groups of phthalates that Deb has put together here, is associated with decreases in lipids in adolescence. So you see that here. So about a 7 to 10% decrease in LDL and in total cholesterol, and then decreases here for MCPP and MEP, only in boys. So we were considering writing a paper which would sort of for girls and their cardiometabolic risk, we just call it POSE. Like we don't know why girls are having sort of higher metabolic risk compared to boys, but this is strengthening that picture. And then in girls, however, with phthalates, so just as we're about to despair, the only effect that pulls up here is actually the one of EHP, but it's the summary of the metabolites also is associated with a decrease in LDL. These are all concurrent exposures, and I think after watching all the BPA studies jump around over the last 3 or 4 years, I'm probably not very confident at all about what we can learn from cross-sectional studies. Because we'd love to be able to cover JAMA, but I don't think that's going to happen. We want to look at it. So the last thing I want to say about this, and then I just have a couple concluding slides, is it's always a risk. You can really polish talk or you bring things you're trying to figure out. And we went with the things we're trying to figure out, and I'm glad I did because I think people in the room may have some ideas or have ideas for us at lunch. So one of the things we were looking at is the association of, actually I think Deb did this slide, but Saturday. Cindy was like, "Where are the slides?" So I think fasting glucose should be out of this title. This is for phthalates and their relationship. I'm sorry. Wait, it really is a new slide. So what this slide shows is what is the relationship between phthalates and glucose. And if you recall from the previous slides, we saw nothing in boys versus girls and nothing overall. But when you stratify by pubertal status, we see something very interesting, which is that we only have effects in pubertal boys and girls. And in boys, phthalates are associated with, in utero phthalates are associated with a reduction in fasting glucose in adolescence once you've gone through puberty. So again, that's when your lean mass has gone way down, or sorry, it's gone way up, and your fat mass has gone way down, and a few other things might have happened in puberty. And then, a little tongue-in-cheek there. And then in girls, the only time we see effect is also after they've undergone puberty, but we see the opposite direction, which is phthalates are actually influencing and leading to higher glucose. So, okay, I think in interest of time, Sue, I won't go into the metabolomics because that's another 4 or 5 slides. So, save that for another talk, and I'll just. What do you want to stay in course, right? Let's stay in course. So, next time we invite you all to come to Michigan or the Children's Center meeting. We'll tell you about metabolomics, and feel free to catch any of us. A number of the slides actually Lisa prepared for us. The bottom line is they're not only fascinating because they're seem to be very labile biomarker. They can change very rapidly in response to diet, for example. But it's also such a new field. It's requiring a lot of very sophisticated statistical approaches as well as laboratory approaches. So we're fortunate that we have a couple of big centers that were funded to look at metabolomics at Michigan. And we're also sort of one of those, be careful what you ask for, because we're like, can somebody look at this? Can somebody look at the mass spec? Who knows about mass spec? What is this compound? And it's so it's kind of a brave new world. So in terms of summary, what we've seen is the effects of the EDCs that we've looked at so far. They really vary, the effects vary by sex, and they vary by the timing of exposure in terms of these cardiometabolic outcomes. Lead exposure seems to have an effect on glucose in boys, but not much else. The HDL cholesterol is not a strong effect. In the metabolomics analysis, it does look like lead is affecting one of the amino acid metabolism in xylose, but we're not sure really what that means. And I think those are the key findings there. We, as we move from a formative center to a full-scale center, we, and I think Susan hinted at this in the New Bedford cohort too, we've been really limited by statistical power, and now that you can start to see the effects are varying by puberty and sex, and in boys and girls, we desperately need more subjects. So we're in the process of expanding our sample. We're very interested in longitudinal trajectories of exposure and outcomes during adolescence. And we did recently get an administrative supplement to look at high-dimensional epigenomics, so genome-wide DNA methylation, and metabolomic responses. And we're bringing in something new called lipidomics, which actually allows you to look in much more detail at lipids. So that's something that I think is very exciting, and I'm hoping will clear up my confusion. So next time you see me, I'll be less confused. And then finally, as I mentioned, Peter actually just last week got this R01 to develop some statistical methods that in about 3 years, we hope we would have software that we could share with the children's centers. The last thing I'll say about that, an example of something he's dealing with is, can you take structural equation modeling and then use high-dimensional data in that? So those are the kind of things we're trying to do, is use very straightforward approaches, but then modify that for high-dimensional data. So this is our group. I won't go through all the details, and I'm going to end on time.